Hello, everyone. Thank you for joining our webinar, Product Packaging Considerations and What to Do When Things Go Wrong. Today's webinar will cover food and ingredient safety considerations and how to select a lab when packaging fails. This webinar is hosted by Dave Riggs and Amber Skaretka of Eurofins SF Analytical. I'm Genevieve Randall, and I'll be moderating this webinar. Before we begin, I'll let you know more about how this webinar will run. The webinar is being recorded, and the slides and recording will be available for you within 3 business days. Our short Q&A session will follow the presentation to answer any viewer-submitted questions. During the webinar, you can submit questions you have using the webinar sidebar menu. Select the Questions tab, type in your question, then hit Enter on your keyboard. Remember, you can submit questions throughout the webinar. If we don't get to your question during the Q&A session, then Amber or Dave will reach out to you individually afterwards. Now I'll give you a quick overview of Eurofins. Eurofins is driven by our mission to contribute to global health by offering the highest quality testing, training, auditing, and consulting services. We strive to listen to our customers and not simply meet but exceed their expectations. Our footprint is global, with 45,000 staff in over 800 laboratories across 47 countries and a portfolio of over 200,000 analytical methods. Eurofins provides a unique range of analytical testing services to the food, pharmaceutical, environmental, and consumer products industries and to governments. All right, Dave, you can start your presentation.
Thank you, Genevieve. It's great to see so many people have tuned in. My name is David Riggs. I'm a 34-year plus industry veteran. I've seen a lot in my years that I hope to share with all of you in an interesting and informative format over the next 35 to 40 minutes. Today we'll talk about the 5 bullet points that you can see on this slide. And then my colleague Amber will provide some actual case studies and also some lab selection criteria that should be helpful as you think about when you're maybe confronted with similar situations for your particular company. And then finally, we'll wrap up with some questions and answers. So the first question is, why should we assess ingredient safety? And there's obviously many reasons to assess both ingredients and packaging safety. And that can start with the integrity of the product, integrity of the package, including compliance with the latest Food Safety Modernization Act guidelines. Could be compliance with regional guidelines, things like California Prop 65, or international guidelines such as REACH, or specific requirements from a country you're exporting to. It also will bolster your products and your company's brand and your company's reputation. And by having a proactive quality, risk mitigation, and testing plan, not only is it going to improve the quality and your product performance, but it's going to give you that control you really need over all aspects of your supply chain, give you the confidence and the confirmation your product label claims, and it should enable you to take increased market share and improve your company's bottom line, which is going to make your boss and make your company's CEO and your shareholders very happy. But most importantly, it's just the right thing to do, in today's modern world where more and more people are concerned about product safety and product quality. So on our next slide, what are some of the other benefits of ingredient testing? Well, reiterating some of the things I said before, not only does the product quality and safety improve your bottom line, but it's going to prevent recalls or getting your company's name in the news or in the media for the wrong reasons. It should help with early detection of product adulteration, fraud, counterfeiting, things of this nature. Also, in the event that there was a legal claim, an insurance claim, or some type of downstream financial claim, by considering all the things we talk about today, you should be able to strengthen your position and minimize your exposure. And again, it's going to give you confidence in what's listed on your product label. And that has a lot of intangible benefits as far as being a good ambassador for your brand across the industry. And it puts you kind of in control and on the offensive, and it causes your competition to become more reactive to where they're going to have to kind of keep up with you. On the right side, I have just a simple table, and this is available from the Food and Drug Administration. They publish this information. But the top 3 reasons for either import detention or product recall, and we kind of amalgamated it together for simplicity. But number 1 is foreign materials and ingredients. Now that includes filth and extraneous matter, which can include, moth artifacts and rat tails and vermin feces, in addition to, glass or plastic or metallic shards. So we kind of amalgamated those together. Number 2 is unlisted allergens, and that list, of course, is growing, just the 8 most common more. And then finally, trace pesticides, trace residues, or heavy metals that are listed above the limits. So you can see those 3 combined are more than 50% of the reasons for product recalls. So if you focus on mitigating those 3, you're going to make a big step in improving your company's quality and mitigating the chances that your company is live at 5 for the wrong reasons. So now we start to dig in, where are these problems encountered? Well, they kind of fall into 2 kind of main categories. Number 1 is contamination of a supplier provided raw material ingredient. That could be chemical residue, could be metal, plastics, glass, and that can come from equipment, packaging, processing, any type of upsets along the way, or it can be from bacterial, so microbiology, fungi. We kind of group those in as pathogens. The second category would be ingredients that are not claimed on the label. Again, allergens, preservatives, additives, adulterants, or other contaminants that are not listed or should not be present. So as we dig a little deeper into the contributing factors for where these problems come from, we like to go upstream because we all want to get to, obviously, root cause analysis, corrective action. So manufacturing and processing failures oftentimes are going to be during the manufacturing or within the packaging steps. And oftentimes it can be from improper treatment, like inadequate hot packing, pasteurization, maybe some type of contaminant that's present from a maintenance that went wrong, or a cleaning in place solution or cleaner that didn't get flushed out properly. And then your packaging failures, oftentimes you'll get extraction, leaching, migration of compounds, and that's really because the material maybe was not designed for an acidic product or a basic product, or you're using it at higher pressures or higher temperatures or lower temperatures it was designed for. The next common failure mechanism is oxygen, moisture, CO2, or light penetrate the packaging, and they cause accelerated failure of the product. So the product tastes good, looks good, it meets all specifications when it goes into the package, but 30 days later, it starts to degrade. And if you have a 6-month or 1-year shelf life, that's going to lead to complaints or potentially recall situations. And then finally, leaks, damage, or exposure to temperature extremes or conditions that were never anticipated when the packaging was specified. And this is becoming more and more of an issue with longer and longer global supply chains. When you made the product, in the same state or the same region, they put it in a package. A day or two later, it was at your distribution center. A day or two later, at the retailer. Now supply chains are coming from Asia, Australia, Eastern Europe. Products may be on a boat for 4 to 6 weeks. They may take weeks to clear customs. They may be in multiple distribution centers as they go up and down our highways and byways around trains and boats. So when you look at planes, trains, and automobiles, there's many, many more chances and much more time for these other external factors to impact your product. So we always want to be cognizant about our longer and more complicated supply chains. So in terms of how we dig into these, one of the tests in our arsenal is chemical verification testing. So in terms of what it is, it's really testing to just verify or determine quality or purity. And typically it's going to be shown on what's called a certificate of analysis, or it's going to be on your product specification. And it may also be for a specific quality designation, and we'll talk about some of those in the later slides. The U.S. Pharmacopoeia is one of the most common ones, but there are many, many others. In terms of, there's four main reasons that's done. It confirms your quality and safety. That's number one. Number two, your purity and content. Next is detection of any contaminants or things that are there that shouldn't be there. And finally, it allows you to either release a product as a raw material or a finished product to whoever your customer is or the next person downstream in the supply chain. So now when we talk about how is the testing performed, again, we break this down again into two more categories. The first category would be compendial testing, and that's according to a recognized method or compendia. Again, the U.S. Pharmacopoeia National Formulary, that's probably the most common, but the Food Chemical Codex is also very common. And then you get to your either region or country specific, where you could have FCC, European Pharmacopoeia, Japanese, British, and ACS, American Chemical Society, AOAC, GB is for China. And again, you sometimes will see things like REACH, or you may see things like California Prop 65, and those tend to be specifics for things like phthalates, heavy metals, or specific toxins. And oftentimes those are a subset of what you'll see in the other compendial methods. And then there's non-compendial methods, and those are typically going to be specified by your client, maybe an industry group, or they could be a method that you've developed with your clients that you've all agreed that this is going to be the criteria for the release of product or acceptance by your customers. In those cases, oftentimes there needs to be method validation, method verification, or some type of technology transfer to make sure that everyone is performing the method according to the same methodology, the same instruments, the same criteria. It's also useful for investigations, the most common of which are foreign material identifications or contaminants. As you recall from the earlier slides, that's where one of the three most common reasons for non-conforming product or product recalls according to USDA and USFDA, but also for investigative problem solving, of which my colleague Amber will talk about in some of the later slides. So in terms of what it's performed on, obviously food and beverage ingredients is the most common area. But in many cases, food and beverage ingredients are also used in pharmaceutical applications. They may not be the active pharmaceutical ingredient, they may be the excipients, so the starches, the colorants, the fillers, the additives that allow the pharmaceuticals to behave differently or have longer shelf lives or be stable in different forms. They also can be used as raw materials or finished products, and oftentimes these might be sold as fine chemicals, or they may be used as and sold in small quantities for analytical standards if the purities meet very stringent requirements. And finally, they're oftentimes used in personal care products, especially in cosmetics, where they can be absorbed into the skin, into the eyes, swallowed, or somehow make their way into humans or animals, or things that are consumed by humans and animals where we're concerned about potential toxicity. In terms of who needs to perform this kind of testing, it's a very broad category. Obviously, the chemical manufacturers, people making these, but also there's a lot of chemical resellers and distributors in our supply chains now, importers and exporters. Obviously, the ingredient manufacturers, in many cases, pharmaceutical companies, medical device companies, cosmetics, and even home health and beauty producers, and in some cases, the retailers. So it's a lot of people are responsible for getting this testing performed or making sure their suppliers are getting this testing performed, and that way they can comply with regulations such as the Food Safety and Modernization Act, California Prop 65, REACH, and various regional and international guidelines. I'm not going to read you all the common tests that are performed. You can see there's a lot of them. But in terms of the ones that are probably the most common, a lot of times the moisture or loss on drying is done because that's indicative of a product maybe was exposed to moisture or packaging failed. Obviously, heavy metals are very, very important. So the big four being mercury, cadmium, arsenic, lead. pH and conductivity are often done because they're fast, easy to replicate, and are indicative of when things start to go wrong. Identification, which is a lot of different ways, but people like fast identification. So a lot of infrared techniques, such as Fourier transform infrared, near IR, very common, or also colorimetric technique, where we take a product, we add a reagent, it turns blue, it turns orange, indicating that it is what we thought. Allergens is very common because, as we said before, it's one of the three most common causes of contamination. Appearance, because it's inexpensive and fast. And then residual solvents and residual pesticides. And then, of course, I say microscopic residue, but just microbiological testing in general is very common because it's one of the most common sources. Microbiology is they're very good at getting in where they don't belong and causing yeast, mold, and bacteria. The good news is most of them are harmless and they have good detection things. So oftentimes we're able to spot those problems before they become serious. In terms of some of the more typical techniques, again, the titrations are real common. There's a lot of different types. Carl Fisher for water, colorimetrics, redox. We use a lot of ovens for things. It's not real exciting, but good for driving off moisture, drying things out, getting them to where we can put them into solution, or ashing off the organics to determine inorganic content. A lot of comparison tubes, so different ampules and crucibles and cuvettes, which can then be inserted in like an instrument to look at things like UV vis. Obviously, different types of titrations. We'll talk about some of the other analytical techniques, the gas chromatography, HPLC, IC, ICP-MS, all your different enzyme assays, UV vis, fluorescence. The list goes on and on and on. So I know in our particular lab, we got over $5 million of state-of-the-art equipment, and there's a lot of labs out there that are much larger than ours. So the amount of instrumentation is mind-boggling and staggering. In terms of some of the other general testing techniques for ingredients, again, pH, color, viscosity, specific gravity, refractive index, and then I talk about TGA and differential scaling calorimetry. Those are thermogravimetric techniques that have really come a long way, and they almost give you a kind of a unique fingerprint that's not of a spectral nature, based upon the thermography of when things melt, when they have phase changes, and very quickly and very cost effectively, you can determine that the product is what you think that it is. When we talk about elemental analysis, this is going to be for most of the things that are kind of on the lower or left side of the periodic table. So, of course, most people are common with all the heavy metals. But we use inductively coupled plasma, and we can do that with either optical emission spectroscopy or mass spec. Optical emission is a little better. It has fewer matrix interferences. With mass spec, it's possible to get down to parts per billion, in some cases part per trillion limits of detection, which is very, very important, especially for excipients or when you start to talk about pharmaceutical type of applications. A very fast technique that's more qualitative in nature, but semi-quantitative, is EDXA or EDS, which is on a very expensive instruments along with a scanning electron microscope. But very quickly we can determine what elements are present, and then we can use ICP or other techniques to really dig down deep and find out maybe where the problem is. Colorimetry, discrete analysis has really come a long way and is a fast, very accurate, very cost-effective method that's being used more and more. And then you get into your ion selective electrodes, ion chromatography. And ion chromatography is especially good for anything that ends in OX. So chlorates, nitrates, sulfates, phosphates, bromates. So anything that have those OXs at the end where there can be multiple different electrical charges of things, ion chromatography is a very powerful technique. So then we get into our specialized techniques, your gas chromatography. Of course, we can match that with flame ionization detection, thermal conductivity detection, which is good for gases, and mass spectral detection, where we can detect over 300,000 different volatile and semi-volatile components. Obviously, liquid chromatography for things that's not volatile. And basically, the rule of thumb there is if it boils at 175 degrees centigrade or less, we're going to use GC. Higher than that, liquid chromatography. But there are some exceptions, and we can do things with derivatization to kind of move from one side to the other. UV absorbance and transmittance is very powerful. We talked about Fourier transfer infrared, near infrared, very fast, very powerful techniques, and they lend themselves to online testing. We can actually put these instruments in your process and get real-time data very quickly. These things can run in literally a matter of seconds and give you real-time information, control your process. UV visible, and then Raman spectroscopy. Raman has some very powerful applications, microplastics, carbon black, certain types of matrices that give FTIR and near IR problems. Raman spectroscopy oftentimes can offer a solution in those cases. And on the right side, you can see just examples. These are the things you oftentimes see on the TV shows, the crime scene and investigation shows where they, figure out that, the butler or the gardener or someone killed them. The only difference is, unlike on TV, it's hard to do those things in an hour. Typical turnaround time is a couple of weeks at the lab, and it's a little more difficult than they make on TV. But we just offered that to try to interject since we're about 15 minutes into the webinar. I want to make sure everyone's still staying awake. Some of the other techniques for the ingredients, we get to ELISA and PCR techniques. Those are really useful for proteins, allergens, yeast, mold, bacteria, fungi, things of that nature. In some cases, we'll have to use light or staining techniques to illustrate these. In other cases, you may have to use fluorescence or polarizing light. In some cases, we may have to gold sputter the materials so we can illustrate them with the scanning electron microscope or transmission electron microscopy. And then at this point, what I want to do is I want to go ahead and turn things over to my good colleague, Amber Skaretka, and she's going to talk about not only the considerations if you're considering a third party to assist with testing, but she's going to share some actual case studies from actual projects we've worked on where we changed the names to protect confidentiality. But I think you'll be able to find these interesting, informative, and really learn so the same things don't happen to you or your company. So with that, I'll turn it over to Amber. Thank you very much.
Thank you, Dave. So yes, I would like to go over the considerations for selecting a lab. In regards to where these tests are being performed, there are a few different factors to consider. First off, you have in-house labs versus external independent labs. You want to evaluate your company's budget, net sales, location, technical expertise, and quality infrastructure for determining if an in-house lab fits your company. If an in-house lab is your path, you'll want to ensure your team is consistently trained with validated methods, that a robust quality assurance program is established, and there's capacity to grow, and very importantly, that there is an unbiased data review. Additionally, it is rare for in-house tests or in-house labs to have lab-specific certifications and accreditations like ISO 17025, as these are very pricey and require a lot of time investment. As such, testing often needs to be sent out annually for verification or by customer request to an accredited lab with accredited methods. In these instances, as well as those not having in-house labs, you almost probably need to utilize an independent lab. Outside, third-party labs are great sources for unbiased, routine, and non-routine net testing. We will discuss more in detail later, but a few important factors to consider are commitment to scientific excellence, including experience and technical credibility, a proven track record of success, a specialized focus on solving obscure and complex problems, and the overall service offerings of that lab. Okay. So in exploring external labs, it is important to ask what services and methods they can offer that fit your needs. Some important questions are to ask, how do they perform sample preparation? Are there multiple methods for isolation and extraction to fit your various matrices? Can they perform analysis by multiple techniques if needed? Is the design of testing or experiments focused on objective and unbiased investigation? Will testing ensure collection of pertinent information, including background data, sample matrix, ingredients added, intended use of product, and regulations surrounding your product? When it comes to methods, it is helpful to know if standardized methods will be used or if modified custom methods are more appropriate. In addition, are these methods validated or accredited? Ask if they will produce qualitative or quantitative data, and what kind of limits can this lab achieve based on the matrix? Lastly, can the lab perform custom method developments and investigative work if your samples do not fit standard methods and sample preparations out there? Further attributes to consider when exploring independent labs are, what are your lab's deliverables? Are they giving you a sample C of A, or do you have a full report? A simple certificate of analysis may be fine for most routine needs, but often a higher level of service is preferred. In many instances, a final report, including rigorous chain of custody details, raw data, relevant spectra, tables and graphs, photos, data interpretation with statistics, method details, and references can be much more powerful than a C of A. In addition, having access to scientists for questions at key project milestones and when reviewing the final report is very useful for understanding work being done and further problem solving. The speed of responsiveness is also a very valuable trait when working with a lab. Considerations for selecting a lab, especially for special projects, is not an easy task, but hopefully today you've been given some information between Dave and I to help you understand where to start. To sum up earlier, questions useful to ask a potential lab could include, is this lab independent, objective, ethical with no conflict of interest? What is their brand reputation and technical ability? Do they service regionally, nationally, or globally? What certifications, accreditations, or licenses do they possess, such as ISO, US FDA, CGMP, USDA registered, or maybe even DEA for controlled substances? What ideally fits your needs is what is the best. Other areas to look at are the lab's years of experience, successful track record, skills in providing solutions when things don't go right, a tentative response rate, sample protocol management, and this can include the documentation, traceability, and storage of your samples, and then overall convenience of using the lab. It is good to insist on confidentiality and client references as well. The right partner should be a trustworthy extension of your company's quality departments, R&D, and technical support functions. So asking specific targeted questions when exploring external lab partners, and if needed, consider an audit if ongoing testing is anticipated. We're now going to describe 6 actual case studies. A few of the details have been redacted to ensure customer confidentiality, but the 6 cases are interesting, informative, and may provide learning lessons to help you avoid something similar happening to your company in the future. In addition, if something like this does happen, you will have some tools for how to investigate the situation and mitigate your company's risk with prompt, effective root cause identification and corrective action moving forward. In our first case study, a fruit juice manufacturer was receiving complaints of off odor and flavor in their product after many years of no problems. They suspected some type of contamination from their process cleaning and disinfection protocol. They collected control samples of a product as well as multiple lots of juice containing the off flavor and off odor, and then submitted samples to Eurofins for analysis. Eurofins scientists attempted multiple sample preparation techniques but settled on liquid extraction and concentration of volatile analytes, followed by headspace GCMS, with comparison of control and suspect samples. As we will see from the chromatogram at the right, there is a red arrow which indicates the suspect's samples and exhibited an unknown peak. Using the mass spectral database, Eurofins scientists were able to positively identify the peak as an active ingredient in the cleaning agent used to disinfect the juice packaging line. The client was able to modify their cleaning and disinfection protocol and eliminate the problem in the future. In the second case study, a commercial mink farm was experiencing high death rates across all demographics of their population, and allegedly the custom formulated feed was contaminated, causing the minks to die off at an alarming rate. Insurance and legal claims were filed against the local feed mill and all of the major ingredient suppliers involved. Investigators observed discrepancies in some of the testing when compared to the evidence found at the mink farm in question. Eurofins was asked to join the investigation to objectively search for the truth and find out what was causing the animals to die. A thorough review by Eurofins scientists identified a number of discrepancies between test results and field observations. It was found that contaminated cardboard barrels originally used for industrial welding materials had been repurposed without adequate cleaning by the mink farm operators and were contaminated with toxic metals and chemicals that were used to transport bulk feed across mink farms, slowly poisoning the animals. A sworn affidavit by Eurofins scientists was reviewed by the judge, and the defendants were found innocent of any claims filed by the plaintiff, and the case was dismissed. In our third case study, a commercial caterer at a convention offered crustacean allergen-free fried rice. Several attendees with food allergies consumed the fried rice, one of whom exhibited a severe allergic reaction and was hospitalized. An astute observer photographed the serving dish of fried rice and collected a sample to be sent to Eurofins for analysis. A positive and negative control samples of fried rice were tested according to ELISA techniques. In triplicate, along with the fried rice collected from the convention, and high levels of crustacean contamination were indeed confirmed. The evidence allowed for a favorable settlement for the plaintiff without excessive legal costs to the defendant's insurer, and the caterer has updated their procedures and training to mitigate the chances of this happening in the future. In our fourth case study, a hot-packed juice product was exhibiting cloudiness a few weeks after bottling, as well as becoming bloated due to excessive gas formation. Microbiological contamination was suspected, and several affected bottles were submitted to Eurofins for investigation. Samples of the juice were carefully harvested and then subjected to specialized isolations, followed by PCR and DNA sequencing to identify the microbial contamination causing the cloudiness and gas formation in the product. A fungus called mucorcercinelloids was identified as the contaminant and traced back to one of the raw materials. Upgraded raw material inspection and acceptance protocols were implemented by the juice bottler, and an online inspection device was installed as a preventative measure, and no further problems have been reported since. In our fifth case study, a large manufacturer of cheese obtained portable water from a municipal water source that had experienced a failure of their carbon bed filtration system. The activated carbon migrated downstream and was undetected for several days, resulting in many tons of non-conforming cheese. The carbon particles later found in the cheese needed to be matched to the failed filtration system, as a large insurance claim was being filed. For preservation of evidence and the way the case was filed, non-destructive techniques were required. Eurofins scientists performed careful isolations and sample preparations on cheese specimens exhibiting the dark contaminant. Scanning electron microscopy with EDXA elemental analysis confirmed the dark particles were indeed activated carbon from the municipal water plant in question. A settlement was reached, and additional filtration and inspection measures were implemented at the cheese factory to prevent any future reoccurrence. And in our final case study, a major fast food chain received complaints of rodent artifacts in several partially consumed chicken sandwiches. The rodent artifacts were confirmed to be present in the baked buns used for the sandwiches, but the fast food chain wanted to know which of their 3 bun suppliers was to blame. Eurofins scientists first harvested portions of the partially consumed buns in question that had not been contaminated with chicken grease, condiments, or any other toppings that would interfere. They then compared the organic acids present in all 3 bun manufacturers to the organic acid profile of the buns containing rodent artifacts. It was found that all 3 bun manufacturers had unique organic acid profile or fingerprint, enabling the offending bun supplier to be identified and corrective action to be taken in order to prevent reoccurrence in the future. In summary, the safety and integrity of ingredients used in food, beverage, and supplement and pharma supply chains is critical to minimize product risk and ensure your brand's integrity at all times. It is important to assess your company's internal and external testing capabilities for quality control, quality assurance, and to understand where any gaps may exist. When considering the use of an external testing partner, using all of the factors discussed today can help make the right choice for your company. There are thousands of labs across the U.S., but only a handful are able to provide independent, competent, and responsive testing and support for a full range of cases discussed today. Be sure to ask the right questions and discuss all aspects of sample submission, method selection, reporting when you're selecting the lab based on your quality needs. Thank you again for turning into this webinar with Dave and I, and we hope that you found it interesting and educational. We will be happy now to answer any questions that you may have.
All right, thank you, Amber. So as Amber said, we're going to move into a short Q&A now. Feel free to continue to submit your questions throughout the session, and any questions that we don't get to at this time will be followed up with individually by either Dave or Amber after the webinar. And with that, our first question is, what should the first steps be if I have a packaging issue with a contract manufacturer?
Thank you, Genevieve. That's a great question. The first thing you want to do is you really want to quarantine and secure the evidence in the site. So quarantine any raw materials, any work in process, any finished product. Collect any and all data from your control systems, log sheets, batch sheets. Get the names and talk to any operators, anyone who has observations who might be able to provide information on what happened and where it happened. So really kind of treat it almost like a crime scene. You want to take photographs or video so you can see, where things were. Was it early in the morning, late at night? Was it cold? Was it hot? Was smoke rolling in from the building next door? In parallel with that, you want to notify your internal quality and your regulatory experts. You don't want to tackle something like this alone. You want to assign a team because you may have to deal with media. You may have to deal with internal stakeholders. You're collecting all of the facts. And in some cases, you may even want to notify your internal or external legal counsel if there is potential for insurance, legal, or significant financial claims.
Thank you. Okay, our next question is, what are the top causes of packaging failures?
Another good question, Genevieve. So as we kind of covered in the presentation, we kind of did it 2 different ways. Typically, packaging failures occur either with a combination of improper packaging materials, some type of damage that occurs in transit, and again, with longer and more global supply chains, there's a lot more places where things can go wrong. Exposure to temperature extremes, which can be very hot, but also very cold. Remember, if things freeze and it contains water, oftentimes they expand, the packaging bursts, then when it thaws, stuff leaks out, microorganisms get in. Those are the most common, but again, heat, oxygen, light, moisture, all those things tend to really be the enemy and contribute to different types of packaging and product failures.
Okay, thank you. That was a good review too. Our next question is, For microplastic particles, do we have a valid method accepted by the scientific community?
Well, I could probably let Amber do that since she's our microplastics expert, but I'll kind of start, and then if Amber wants to chime in with some specifics. So Eurofins is able to perform microplastic analysis in bottled water and drinking water and different water matrices by both the SUNY Fredonia method, which is accepted in the industry, and by the NOAA, though that's the National Oceanographic Association method. And we also are developing a proprietary internal method that uses micro FTIR. But there's a lot of work involved with that. So I don't know, Amber, is there anything you want to offer on that since you are a bit of a specialist in the microplastic space?
Yeah, sure. So with microplastics, there isn't any kind of thresholds or limits established. Right now, I would say industry-wise, we're all just trying to figure out how to bring out validated methods to be able to actually test these particles, because there's a lot of interference. There isn't really, it's really hard to determine if the analysis or instrument is indeed analyzing just plastics instead of something else. So there's still a lot of research going on, including at Eurofins lab. So there isn't anything, yes, established right now. It's very difficult, but it's coming, and it's coming quickly.
The thing I would add to what Amber said is there's a lot of interest as far as microplastics in dairy matrices. Obviously, milk, a lot of people consume it. In some cases, fed to children. And also, there's been a lot of media coverage in the last several weeks about maybe plastics are migrating or leaching artifacts from tea bags. So there's been a lot of media coverage for those two. So those two tend to be related to microplastics in water, because obviously milk is primarily an aqueous matrix with some fats, some proteins, other additives, and obviously tea bags, because tea is consumed around the world in large quantities, and there is concern about microplastics in those particular beverage matrices.
Thank you. Our next question is, all right, is the LOD test the same as percent moisture?
The answer is no, although they're related. So loss on drying is going to measure anything that's volatile that, well, let's just say it's an LOD at 105 degrees centigrade. Things like ethanol or methanol or acetone, those solvents will also evaporate at those temperatures. And of course, moisture will as well. So loss on drying is anything that basically boils off or evaporates at those conditions, versus moisture is specific to just H2O or water. So in many cases, we'll run a loss on drying, but then we may also perform something like a Carl Fisher titration, which is very accurate and very precise for low levels of moisture. And even when you do an LOD, it is possible for some water to be present, because water can bind to the matrix and it can form hydrated forms, or it can still be present in low concentrations, even above 100 degrees centigrade, depending on what the sample matrix is.
All right, thank you. Our next question, what are the most common causes of ingredients going bad prematurely?
Yeah, we kind of hit on that before with the packaging failures. So obviously improper packaging will cause ingredients to fail. And that's why it's important, if you're changing packaging, you want to perform stability testing. So expose your products to a 40 degrees centigrade, 75% relative humidity, because in 3 months' time, you can confirm a 1-year shelf life. So product stability testing, very important. And then temperature abuse. And that can be both hot temperatures, cold temperatures, but also humidity extremes. So a product might just fine if it's stored in a dry, cool environment like the desert, but then if it goes into a more like a rainforest environment, the moisture will penetrate through the packaging if it doesn't have a good moisture barrier. Exposure to oxygen. Oxygen does just that. It oxidizes materials. And it's very insidious in that it can pass through what appears to be impermeable membranes. A lot of people think, oh, it's packaged in plastic, so water and moisture and CO2 can't get through. Incorrect. A lot of polymers can be permeated by water, by oxygen, by CO2, by light. That's why oftentimes packaging materials might have 5 or 7 plies, including moisture and different types of vapor or light barriers. But again, improper packaging materials, some type of physical damage in transit due to long supply chains, exposure to temperature extremes, hot or cold, and oxygen or moisture contamination. Those tend to be the most common contributing to the packaging failure. So you can see, packaging and those different types of exposure elements really go hand in hand, and they have a synergistic effect to really accelerate the severity of failures and also increase the likelihood of failure. So I encourage everyone tuning in today, take a look at your products, take a look at your supply chains. What kinds of things can you do internally to mitigate the chances of those types of agents to degrade or impact the quality of your product or your package?
Okay, thank you. And this is going to be our last question for today. Remember, if your question hasn't been answered or you haven't asked your question yet, then Dave and Amber will follow up with you, or you can also contact them at the email addresses on the screen, or you can contact their lab, Eurofins SF Analytical, with any other questions. And our last question is, if we establish SOPs, to what extent do our SOPs need to be verified? Would every device used need to be traceable to some national standard or calibration?
That's an excellent question, and probably I'm not going to be able to answer it in completely today for all circumstances. My advice to people is, if it involves human beings, train it or document it in your SOPs. If it's a instrument or something you plug in or something you measure with, calibrate it. And so depending on whether you're ISO certified or you're GMP lab or other different quality management systems and criteria that may be industry specific or you establish with your downstream customers, will determine, does everything really need to be inspected and calibrated? In many cases, the answer is no. I tell people, err on the side of simplicity. Just a simple set of procedures that say what you do what you say, inspect, maintain, calibrate your instruments, and make sure that whatever testing you're performing is fit for use, fit for the matrix involved. That's going to prevent 99% of the problems. But if you have more detailed questions with that, again, I would encourage you, talk to whoever is your director or manager for quality within your own organization. And by all means, you're welcome to reach out to Eurofins. We've got lots of smart people. We'll be happy to tell you about every different type of regulation around the world, known to men and women, and give you advice as to whether any additional auditing, inspection, or certification is warranted.
Okay, thank you, Dave. And with that, that wraps our Q&A session. I want to remind everyone that a recording of this webinar, along with a copy of the slides, will be available to you within 3 business days, so by Friday. Thank you, Dave Riggs and Amber Skaretka, for sharing your presentation and expertise. And thank you, everyone, for attending.